Warning: the following video may not be appropriate for children under the age of 1.5 months or teachers who are easily offended. No tables were harmed in the making of this film. The producers of this film would like to thank the aliens who will one day take over planet Earth and turn everyone into bananas for not destroying this footage. The angry doorknob has been tracked. No animals were harmed in the making of this film. I see you.
Good afternoon. Claire? Here. Ellie? Here. Kurt?
Here.
Sheila? Here. Andy?
Here.
Kira? Here. Phineas?
Yep.
Verb?
Here.
Get out your notes, eh?
Eukaryotic cells are thought to have evolved from prokaryotes living in larger prokaryotes. This is called primary endosymbiosis and is supported by structural and functional similarities. Heterotrophic eukaryotes evolved first without chloroplasts. Autotrophs arose from heterotrophic eukaryotes that had involved autotrophic cyanobacterium. The autotrophs then had a selective advantage from having a cyanobacterium. Green and red algae, after being ingested by heterotrophic eukaryotes, became endosymbionts. Originally enclosed in food vacuoles, the algae would have become organelles. Both their presence and self-replicating abilities helped the host. Secondary endosymbiosis is the term given to this type of process, in which autotrophic eukaryotic protists became endosymbiotic in a heterotrophic eukaryotic protist. Protist diversity relies on secondary endosymbiosis. For example, green algae became euglenozoans. They have the potential to be both autotrophic and heterotrophic depending on the presence of sunlight. Within the red algae groups of dinoflagellates and stamenophores, species may be exclusively autotrophic or exclusively heterotrophic. Another group from red algae are apicomplexans, who require one of the organelles they acquired by secondary endosymbiosis. They are all parasitic. The chart you can find on page 331 may also be helpful. As you can see,
This is weird. Hey, where are we? Protists. Protists are an extremely diverse assortment of eukaryotes. I know! We're in the textbook. Well, at least it's better than a classroom. Come on, let's explore.
protists are a collection of diverse unicellular eukaryotes. If a protist is autotrophic and produces energy by photosynthesis, then it is known as algae. If they are heterotrophic, they eat bacteria and protists and are known as protozoans. Most protists live in aquatic environments. Endosymbionts live inside a host organism. Symbiosis is the association of two or more species. Protists are the simplest of eukaryotes, although their cells are very elaborate. They have a membrane-enclosed nucleus and a specific pattern of microtubules.
Oh, cool! I've never seen a place like this before. I think these are alive. Ah! That's it. Ferb, I know what we're gonna do today. I know what this looks like. This looks like a...
You all signed the waivers, right?
Perry, your next assignment requires an in-depth knowledge of bacteria. As such, we have hired an expert to explain the details. I'll turn the comm over to her.
Hi, Cherry. It's an honor to meet such a high-placed agent. Now, bacteria are a very diverse group, but they may be divided into 9 categories. 5 of these groups are subgroups of a clade of gram-negative bacteria known as proteobacteria. The subgroup Gamma proteobacteria includes food poisoning causing bacteria such as Salmonella and E. coli. These proteobacteria are photosynthetic and may live in animals' intestines. The second bacterial group is chlamydias. They live in host cells, and one of its constituents causes blindness and a common STD. Spirochetes are helical and move with internal filaments that rotate for movement. Particularly noticeable spirochete pathogens cause syphilis and Lyme disease. There are also gram-positive bacteria. The subgroup actinomycetes help to decompose organic matter in the soil. These bacteria are also used by pharmaceutical companies as antibiotics. Included in gram-positive bacteria are mycoplasmas, which are the tiniest cell yet discovered, with a diameter of only 0.1 micrometer. Finally, cyanobacteria are prokaryotes with photosynthesizing capabilities. Today, they provide food for aquatic environments, but historically, they were instrumental in the process that made our atmosphere aerobic. That's all the information I have for you, Cherry. Good luck!
So, Cherry the chinchilla, you have come to the extreme environments that Archaea love. Nothing can defeat them. I have made them my allies, so I will be invincible. Allow me to introduce you to them. Cherry the chinchilla, these around you are extreme thermophiles. They can live here, near geysers, or in deep ocean vents that are boiling hot. By oxidizing sulfur, they can survive, just like I will, against you and your agency. And if you destroy my friends, the thermophiles, I'll just go to their cousins, the extreme halophiles. They live in water with up to 20% salinity, more than 15% greater than seawater. I would have gone there first, but my doctor advised against the presence of too much sodium. Or there is methanogens, who live in anaerobic environments and give off methane. I would have made their environment my headquarters, but they like living in mud and animals' digestive tracts, which is disgusting. Now, I will show you my next great plan. Last Friday, I wanted to buy a pot holder for my mother. I love her so much, but when I went to the store, there were too many people and the lines were so long. It occurred to me, if I found a way to destroy all these people, I wouldn't have any trouble buying that pretty pot holder. I came to the conclusion I'd need to use either an exotoxin or an endotoxin, because they are the poisons produced by illness-causing bacteria. Here's an exotoxin producer, Staphylococcus aureus. This can cause skin to slough off, toxic shock, or vomiting. To cause these effects, bacteria cells secrete poisonous proteins. Endotoxins, on the other hand, are components of the outer membrane of gram-negative bacteria that are released when the cell dies or is digested. They cause fever, aches, and sometimes septic shock. Now, you may ask how I shall utilize this information. I want to clear out as large a population as possible of people. Anthrax would be perfect. Creating a weapon with anthrax means preparing air-dispersing endospores. When the shoppers breathe in, an exotoxin will increase the lethal levels in the blood. Plague is another option. After it has been transmitted by fleas, people will develop swellings and black blotches. In either blood or the lungs, the plague is deadly. My final idea is to use botulinum, the most deadly poison on earth. It paralyzes the muscles required to breathe by blocking the nerves that cause contraction. If I use all three, no one will stand between me and my potholder.
You're killing me, kid.
Many prokaryotes are beneficial to us. They are an important part of the chemical cycles of nitrogen and decomposition. Bioremediation is used to remove pollutants from soil, air, or water. In sewage treatment, solid waste is separated and added to anaerobic prokaryotes. These bacteria and archaea decompose the sludge into fertilizers. The liquid waste that had been previously separated are filtered over rocks and aerobic bacteria to sterilize them. Fertilizers were created to do the work of soil-eating bacteria in order to clean oil spills. Bacteria may also be used to clean mining drainage. The drainage water is acidic and poisonous, but prokaryotes, like the thiobacillus, help stop the contamination. In the actual mining process, microbes can be used to extract precious metals.
This has been wrong on so many levels.